Hello, my name is Ciara Duggan and today I will be presenting Microplastics in Dragonflies, which is based on the thesis that I submitted as part of the Bachelor of Science degree at NUI Galway, and my supervisor was Dr. Michelle Duggan. So firstly, what are microplastics? Microplastics are plastics which are between 1 to 1,000 micrometers in length. Plastics themselves are synthetic organic polymers which are typically manufactured using petroleum. Microplastics can be classified as primary or secondary microplastics. Primary microplastics are produced at that scale for a specific purpose, for example, microbeads and exfoliants. Secondary microplastics, on the other hand, arise as the result of the degradation of a larger plastic particle, for example, a water bottle. So why did we choose to study dragonflies? We chose to study dragonflies because they're top freshwater predators. Dragonflies are semi-aquatic and they have 4 main life cycle stages: the egg, the prolarva, the larva and the adult. We chose to focus on the larval stage. The larvae are ferocious top predators. They almost have 360-degree vision and they have a hinge-like mouthpart appendage. Another reason we chose to study larvae was because they were shown to be useful bioindicators in a study published in 2014, which studied mercury bioaccumulation in dragonflies. Aims of this study. The first aim of this study was to investigate the presence of microplastics in the digestive tract of dragonfly larvae. The second aim of this study was to quantitatively compare the presence of microplastics found in specimens of both a rural and urban habitat. The rural habitat which we chose was Ross Lake in County Galway, and the urban habitat which we chose was Eglinton Canal, found in the city of Galway. Sample processing and analysis. Once samples were collected and preserved, they were then dissected. The crop, which is essentially the stomach of an insect, was removed. This tissue was then digested using proteinase K. Nile red was then added to any remaining material and observed under a fluorescent microscope. Image day was used to quantify any microplastics present. Contamination prevention was essential throughout this experiment, so a cotton lab coat was worn and pipetting intervals were limited. Results. On average, there was 0.7 more fluorescent particles in Eglinton Canal samples compared to Ross Lake samples. However, when this was statistically analysed, it was shown that there was no significant difference between the sample areas. It was also shown that there was a low variance throughout this experimentation in the blank results. This shows that contamination rates were consistently low. Discussion and previous studies. It was predicted at the beginning of this study that there would be a higher microplastic quantity in dragonfly specimens from the urban habitat. However, this was not the case. This suggests that there is a similar plastic contamination rate in both areas. Likely sources of plastic pollution within Ross Lake include peat extraction, illegal dumping, and the loss of recreational fishing gear. During peat extraction, plastic bags are typically used to both store and transport turf. So what can happen is these plastic bags can end up in the environment and degrade, entering our waterways. It is also more than likely that the plastic within these dragonflies originated from their prey items. A study done by Cuthbert et al. In 2019 showed that there is a positive correlation, between predation and the trophic transfer of microplastics. Implications of microplastic pollution in freshwater habitats. Microplastics pose a potential global health threat. They have the ability to carry both toxins and pathogens. A pathogenic fish bacterium has even been found on microplastics present in the North Adriatic Sea. Humans ingest 5 grams of plastic a week, which is equivalent to the size of a credit card. Findings of this study and future studies. This is the first study to demonstrate that microplastics are found in the digestive tissue of dragonfly larvae. It also shows that they have the potential to be used as microplastic bioindicators. I believe that future studies should focus on microplastics present in other areas of dragonflies, for example, adipose tissue. I also believe that the correlation between microplastics found in dragonfly larvae and dragonfly adults should be compared. Conclusion and mitigating plastic pollution. I believe that an integrative approach is needed in this area of study. This will ensure that all areas and all organisms are being represented. Through innovation, reducing plastic production, and also using an integrative waste management system, it will be possible to mitigate this issue and prevent plastic ending up in the environment in the first place. Thank you for listening, and also to all those who helped throughout this project.